Hello, my name is Michael West. I'm the CEO of Biotime. Biotime is a biotechnology company focused on the emerging field of regenerative medicine. And today I'm going to be talking about one aspect of that field, technology called FateSpaceScreening, developed by scientists here at Biotime. I'll be making some forward-looking statements that have associated with them certain risks and uncertainties, and we refer the viewers to our filings with the SEC for more detailed information about the company. The foundations of regenerative medicine were laid in the 1990s with the discovery of human embryonic stem cells. For the first time in the history of medicine, scientists had the ability to manufacture on an industrial scale all of the cellular components, building blocks of the human body. This is in part due to the fact that these cells are unique in the history of medicine, not only in regard to their ability to make all the cells of the human body, but in their ability to proliferate without limit, a property we call cellular immortality. And this is in part due to the fact these cells come from what is called the germline. It's a lineage of cells that connects the human species from one generation to another. So they have the remarkable property of maintaining high levels of telomerase, an enzyme that allows cells to proliferate without aging. It is now even possible to take cells from an aged human, say skin cells, and using transcription factors, coax those cells back in time, sort of a cellular time machine, back to this embryonic state. These cells are called induced pluripotent stem cells, and they have the advantage of being essentially identical to human ES cells, but since they were made originally from a cell of the human body, an existing patient, let's say, they are genetically identical to that patient and would not expect it to be rejected from the patient, and can be used in a wide array of age-related degenerative diseases to replace dysfunctional cells of, say, the brain in Alzheimer's or the heart after a heart attack with functionally young cells to regenerate tissue function associated with aging. The significance of these new scientific discoveries is that we're entering an era in the United States known as the age wave, a gradually accumulating number of aged people associated with the aging baby boom population. And with the problems of aging comes the most critical challenge to our society, and that's the mounting number of chronic age-related degenerative diseases that have costs associated with them that are just enormous. So the intensive care required to take care of a person with Alzheimer's disease, for instance, or the physical therapy associated with hip replacement and osteoarthritis. The promise of regenerative medicine is the development of new technologies that could repair and replace the cells of an aged tissue and not merely ameliorate the problems associated with aging with the drug approaches, which oftentimes are not an effective cure. The challenges biotechnology companies have faced in using these cells to manufacture the diverse cells of the human body are shown in this figure. At the top we see the first generation approaches, which were to take these pluripotent stem cells, whether they be human ES cells or iPS cells, and to expose them to growth factors that shift the cells toward the desired cell types. The problem with these approaches is that there are so many decisions that cells have to make in becoming a heart muscle cell, let's say, from an embryonic stem cell, that these cells often drift in an unpredictable manner, leading to impure and poorly characterized cell formulations for human cell therapy. At the bottom we can see the approach taken by Biotime scientists. We call this PureStem technology. Here we take human embryonic stem cells in this example and push them downstream into what are shown here as red arrows that we call human embryonic progenitor cells. These are cells that are no longer an embryonic stem cell and are not yet a fully differentiated cell type of the human body, but are somewhat intermediate, and so we call them embryonic progenitors. A unique feature of PureStem is we take these cells starting with one cell, one embryonic progenitor cell, and expand it into a vast number of cells, a cell line, all of which are identical. so we call them clonal embryonic progenitors. Pure stem cell lines have numerous advantages over previously described manufacturing technologies. First, we've been able to demonstrate that there are over 200 cellular components of the human body that can be scaled in this manner. Secondly, the cells are at a purified state that, to our knowledge, is unsurpassed in the industry. By purifying cells from a single embryonic progenitor cell type, we've essentially eliminated all the other branch points of the developing human body. Thirdly, the cells are easily scalable, as you can see in this video of an example of progenitors from the developing human jaw. These cells propagate and proliferate exponentially, doubling it approximately every day so that we could make hundreds of millions of doses of many of these cell lines to treat the degenerative problems of aging. The novelty of the PureStem technology has allowed us to file for patent protection on both compositions of matter, since we believe we were the first to isolate many of these cell types. The U.S. Patent Office can grant composition of matter claims to such inventors, and methods for the manufacture and scale-up of these products. And lastly, novel means of turning these PureStem progenitors into the desired cell types useful in medicine. But how do we discover how a novel cell type, like a clonal embryonic progenitor, can become the final cell type of the human body? Let's say, for example, the cartilage of the knee or a developing tooth. This is what we call FateSpace screening, a novel technology invented by Biotime scientists. This slide shows how FateSpace screening works. Biotime scientists take these greater than 200 progenitor cell lines, expose them to a wide array of diverse differentiation conditions, such as the growth factors we show here, and then the expression of the genes in these cells are obtained by exposing the RNA from these cells to gene expression microarrays, generating now over 100 million experiments or data points on gene expression in these novel cell types. Our challenge historically at Biotime has been, what do we do with this sea of data, over 100 million experiments demonstrating the speed at which scientific discovery can be done in the modern era. Shown here is a gene expression heat map of some of these data points, displaying for the first time to the scientific community what gene expression looks like in these stem cells, important markers of these cells that can be used in the manufacture of new products. In our collaboration agreement with our subsidiary, LifeMap Sciences, we're sharing this resource of over 100 million data points, and LifeMap scientists are using proprietary computer algorithms to predict the matching of these cells with cell types of the human body, what's called cell matcher, and with protocols for differentiating these cells into various cell types, or protocol predictor. Our collaboration agreement with LifeMap Sciences regarding FateSpace screening is part of our strategy to lead in the commercial development of pluripotent stem cell technology in the emerging field of regenerative medicine. Thank you very much.